On a September morning in 1928, Alexander Fleming looked down at a petri dish he had almost certainly ruined, and instead of scraping it into the disinfectant bin, he reached for a lens and looked closer. The dish was one of dozens left behind on a cluttered bench at St. Mary's Hospital in Paddington, London, while Fleming spent most of August on holiday in Suffolk, and it now held a blot of blue-green mold surrounded by a clear ring where his bacteria had simply stopped growing. The question worth asking is not whether Fleming was lucky, which he clearly was, but why this particular untidy, quietly stubborn bacteriologist was the one standing there willing to notice it. Fleming had a reputation among colleagues at St. Mary's for a chaotic bench and a taciturn manner, a man who published sparingly, spoke in short sentences, and preferred experiments to arguments. He had trained under the immunologist Almroth Wright, absorbed Wright's skepticism towards some antiseptics, and spent years patiently studying how bacteria behaved rather than chasing quick results. That patience mattered on this particular morning. The mold, almost certainly Penicillium notatum, had likely drifted up a stairwell from a mycology lab one floor below, settling onto a culture plate of Staphylococcus that Fleming had simply forgotten to put away before leaving town. He did not have a Hollywood epiphany. He grew the mold, extracted a crude broth he called mold juice, and published a modest paper in the British Journal of Experimental Pathology in 1929, coining the word penicillin along the way. It attracted almost no attention. Fleming was a bacteriologist, not a chemist, and he could not purify or stabilize the active substance enough to prove it could work as a medicine in a living body. He kept the mold strain alive for years afterward, mostly as a laboratory curiosity he liked to show visitors, and moved on to other projects. The organism that would eventually save millions of lives spent over a decade as an unfinished footnote in one man's notebook. It took a very different kind of personality to turn it into medicine. At Oxford's Sir William Dunn's School of Pathology, the Australian pathologist Howard Florey assembled a team built for grinding systematic work, anchored by the biochemist Ernst Chain and a resourceful, self-effacing researcher named Norman Heatley. Where Fleming had improvised and set the problem aside, Florey's team industrialized it, purifying penicillin, testing it on infected mice in 1940, and by February 1941 treating a policeman named Albert Alexander, whose infection had spread from a rose-thorn scratch. Heatley, lacking proper laboratory glassware in wartime Britain, rigged extraction apparatus out of bedpans, biscuit tins, and milk churns because there was nothing else to use. Even industrial-scale rescue arrived by accident. Manufacturing penicillin in bulk required a far more productive mold strain than Fleming's original sample. And American researchers at a Department of Agriculture laboratory in Peoria, Illinois, founded in 1943 when a lab assistant named Mary Hunt, nicknamed Moldy Mary for her habit of bringing in rotting fruit from local markets, spotted an unusually rich golden mold growing on a cantaloupe. That strain became the ancestor of most industrial penicillin production for years afterward. When the 1945 Nobel Prize in Physiology or Medicine went jointly to Fleming, Florey, and Chain, Fleming became the public face of the discovery, while Florey privately resented a mythology that credited one man's overlooked dish more than the years of unglamorous chemistry, animal testing, and wartime scrounging that actually made it a medicine. Heatley, whose improvised apparatus made the Oxford work possible, received no Nobel recognition at all, though Oxford eventually granted him an honorary doctorate of medicine in 1990, the first ever given to a non-physician there. A similar pattern, an anomaly noticed rather than dismissed, had already reshaped physics 3 decades earlier in Paris under a sky that simply would not clear. Henri Becquerel, heir to a family line of physicists stretching back to his grandfather Antoine César, was investigating whether uranium salts known to phosphoresce might also emit the newly discovered X-rays after exposure to sunlight. In late February 1896, he wrapped photographic plates in black paper, placed uranium salt crystals on top, and waited for sunlight that Parisian weather refused to provide for days on end. He put the wrapped plates and salts away together in a drawer, and out of habit more than expectation, developed them anyway on the 1st of March. The plates were fogged dark, strongly exposed, despite having sat untouched in darkness the entire time. Becquerel's own hypothesis that sunlight-triggered phosphorescence was somehow the source collapsed on contact with his own evidence, and he recognized quickly that the uranium salts were emitting something on their own, with no external energy source required at all. He reported the finding to the French Academy of Sciences within days. Becquerel was careful and thorough, but not a man eager to build an entire new field from his own discovery. He largely handed the mystery onward. It landed on a young Polish-born doctoral student named Marie Curie, working with her husband Pierre in a converted shed at the School of Physics and Chemistry in Paris that was, by her own later description, freezing in winter and stifling in summer. She systematically tested one mineral after another, coined the word radioactivity to describe the phenomenon, and found that a uranium ore called pitchblende was far more radioactive than its known uranium content could explain. That discrepancy, chased down through backbreaking processing of tons of ore, led to the isolation of two new elements, polonium and radium, in 1898. Becquerel shared the 1903 Nobel Prize in Physics with Pierre and Marie Curie, an honest accounting of a discovery that began with one man's accident and was completed by two people's exhausting, deliberate labor. Marie Curie died in 1934 of a blood disorder now understood to be connected to decades of radiation exposure. Her laboratory notebooks remain radioactive enough today that they are kept in lead-lined boxes at the Bibliothèque Nationale de France. Radio noise rather than photographic fog delivered the next accidental confirmation of how the universe itself began, this time inside a Bell Telephone Laboratories horn antenna in Holmdel, New Jersey in 1964. Arno Penzias and Robert Wilson had access to a large, extremely sensitive antenna originally built for satellite communication work, and they wanted to use its downtime for radio astronomy. Instead, they kept picking up a faint, persistent microwave hiss that refused to go away, present in every direction they pointed the dish, at every hour, in every season. Being careful instrumentalists rather than theorists, they assumed the fault was mechanical. They checked for interference from nearby New York City, rewired connections, and in an episode now retold so often it has become almost folklore, climbed into the antenna to scrape out pigeon droppings and evict a resident pair of pigeons, suspecting the bird's waste might be reflecting stray signals. The hiss barely changed. What Penzias and Wilson did not know was that 30 miles away at Princeton, physicist Robert Dicke was leading a team including James Peebles, Peter Roll, and David Wilkinson, who were actively building their own detector to search for exactly this signal, a theoretical afterglow predicted to survive from an early, searingly hot universe. A phone call between the two groups settled it within weeks. Dicke is remembered by colleagues' later recollection telling his team plainly that they had been beaten to it. In 1965, the Astrophysical Journal published two papers side by side, one from Penzias and Wilson simply and cautiously reporting an unexplained excess of microwave noise, the other from Dicke's group explaining what it meant. The first direct observational evidence for the Big Bang, Penzias and Wilson received the 1978 Nobel Prize in Physics alone, an outcome that left a lingering sense of imbalance in the physics community. Since Dicke's team had done the theoretical work and had been actively racing to make the very measurement that landed, instead on someone else's rooftop antenna. Not every accidental breakthrough arrived through cosmic patience. Some arrived through a chemist's mildly wrecked afternoon. In 1942, Harry Coover was working at Eastman Kodak's laboratories in Kingsport, Tennessee, on a wartime project to develop clear plastic gun sights, testing a family of chemicals called cyanoacrylates. They were useless for the job, since they stuck to absolutely everything they touched, ruining lens after lens, and the project shelved them as a failure. Coover moved on, and for nearly a decade the substance sat quietly out of favor. In 1951, now supervising research at Kodak's Tennessee Eastman division, Coover assigned a chemist named Fred Joyner to test heat-resistant polymers for jet aircraft canopies. Joyner smeared a cyanoacrylate compound between two lenses of a refractometer to measure its optical properties and could not pry the instrument apart afterward. Where an earlier version of Coover had seen only a nuisance, this time he recognized the same stubborn stickiness as the actual product. Kodak patented and marketed it in 1958 as Eastman 910, later known worldwide as Superglue. Coover became something of a showman for his own invention, once famously lifting a television host off the studio floor with a single drop during a live demonstration, and went on to hold well over 400 patents before his death, receiving the National Medal of Technology and Innovation in 2004, partly for the compound's later use sealing battlefield wounds during the Vietnam War. A wrong resistor produced an equally quiet transformation in medicine 4 years later in a barn workshop outside Buffalo, New York. Wilson Greatbatch, an assistant professor at the University of Buffalo, was spending unpaid evenings building a device meant to record irregular heartbeats for two local surgeons, William Shardak and Andrew Gage, at the Buffalo Veterans Administration Hospital. Reaching into a box of components in 1956, he pulled out a resistor of the wrong value, wired it into his circuit anyway, and watched the device begin producing a rhythmic electrical pulse instead of recording anything, on for a fraction of a second, off for about a second, a pattern that echoed the timing of a human heartbeat almost exactly. Greatbatch recognized instantly what earlier pacemakers, bulky, wall-powered, and unusable outside a hospital room, could never provide: a small battery-driven device that could be implanted inside the body itself. He set aside the project he had actually been paid to build and spent roughly 2 years and his own savings perfecting the idea, testing it on dogs with Shardak and Gage before the first human implantation in 1960. That early patient survived 18 months, limited chiefly by the battery technology of the day, a constraint later solved by corrosion-resistant lithium batteries that pushed device lifespans past a decade. Greatbatch went on to hold roughly 325 patents and later insisted in interviews recounting the story that the wrong resistor supplied the spark, but that the 2 years of unauthorized, unpaid nights in a barn supplied the actual invention. Accidents also hide in drawers that nobody has opened in decades, as the paleontologist John Ostrom discovered while excavating in the Clovelly Formation near Bridger, Montana, in 1964. His Yale field crew, including his student Grant Meyer, uncovered the bones of a lightly built predator with unusually sharp, curved claws, an animal that looked nothing like the slow, dim-witted reptiles standard textbooks still described. But the deeper accident had already happened decades earlier, and 30 years' worth of dust away, in the specimen cabinets of the American Museum of Natural History in New York. Searching through old collections for comparison, Ostrom found that the legendary fossil collector Barnum Brown had dug up bones of the very same animal in the same Montana beds back in 1931, 33 years before, and had simply never gotten around to describing them. The specimens had sat mislabeled and largely unstudied for a generation. Ostrom combined Brown's forgotten material with his own new finds and formally named the animal Deinonychus antiropus in 1969, describing its stiffened tail, grasping hands, and above all a retractable sickle-shaped claw on an agile hind foot, built for speed and active predation. The description helped ignite what paleontologists later called the dinosaur renaissance, reviving a 19th century idea, traceable partly to Thomas Huxley, that birds had descended from dinosaurs rather than some separate reptilian ancestor. Ostrom himself remained a cautious, methodical scientist, allergic to overstatement, while his own student Robert Bakker pushed the same ideas into far louder public territory, creating a quiet tension between a careful discoverer and a more theatrical popularizer who often spoke with more certainty than his mentor allowed himself. Ostrom later found further confirmation not in a new excavation at all, but in a Dutch museum drawer, recognizing in 1970 that a fossil catalogued since 1855 at the Taylor Museum in Haarlem as a pterosaur was in fact an Archaeopteryx, a genuine transitional bird dinosaur specimen that had simply been misidentified for over a century. The most recent entry in this lineage of accidents came from a patch of ordinary dirt outside a plastics recycling facility in Sakai, Japan. In 2016, Kohei Oda at the Kyoto Institute of Technology, working with researchers including Shosuke Yoshida, ran a student-led screening project searching local sediment for bacteria capable of breaking down PET, the tough plastic used in most disposable drink bottles. Among hundreds of samples, one previously undescribed bacterium was found actually feeding on PET, secreting 2 enzymes in sequence that dismantled the plastic's polymer chains into simpler compounds it could digest and use for growth. Nobody in the lab had engineered this organism. It had evolved that ability on its own, in the wild, from ordinary environmental bacteria that had only a few decades of exposure to a synthetic material invented in the 1940s. The team named it Ideonella sakaiensis, after the town where they found it, and published their results in the journal Science in 2016. The story compounded almost immediately. In 2018, Researchers at the University of Portsmouth, led by John McGeehan, were simply trying to understand the enzyme's structure by tweaking it slightly, and accidentally produced a mutant version that broke down PET even faster than the natural original, an accident layered directly on top of the first one. By the early 2020s, engineered descendants of that enzyme were being tested at pilot scale for plastic recycling, still far from solving the global scale of plastic pollution, but treated seriously as a genuine tool rather than a novelty, born from a soil sample nobody expected to matter. Across a century that runs from a cloudy week in Paris to a patch of contaminated soil in Sakai, these stories share less a lack of skill than a shared refusal to throw the anomaly away. Fleming's dish, Becquerel's fogged plate, Penzias and Wilson's stubborn hiss, Great Batch's mismatched resistor, Ostrom's forgotten drawer of Barnum Brown's bones, all rewarded people willing to stop and ask why the result did not match the plan, rather than simply discarding it and starting again. Louis Pasteur is often credited with the observation that chance favors the prepared mind, a line quoted so often it has become nearly a cliché, yet the archive backs it up more than most clichés manage. What the archive also shows, less comfortably, is that credit rarely distributes itself as evenly as labor does. Fleming became a household name while Florey, Chain, and Heatley did the harder work of turning his mold into medicine. Becquerel opened radioactivity while the Curies paid for its full unraveling in tons of ore, and in Marie's case, in her own declining health. Dickey's team predicted the very signal that Penzias and Wilson happened to be the ones holding an antenna for. Accident supplied the opening in every one of these stories. But it was character, patience, rivalry, and often somebody else's unglamorous follow-through that decided what the accident was allowed to become.